Hi everyone, my name is Anna and I'm a cephalopod neuroscientist at the University of Cambridge. Different from more traditional neuroscience model systems like mice, fruit flies, non-human primates, or nematode worms, my research uses octopus, or octopi, or octopuses, all are grammatically correct, as our model organism. I, like many other neuroscientists who work with cephalopods, am super excited about all of their potential as a model animal in neuroscience research. Coleoid cephalopods are a subclass of animals from the mollusca phylum that includes octopus, squid, and cuttlefish. These marine critters are commonly known for their interesting behaviors and maybe for being a bit tasty, but they also have really intriguing complex nervous systems and intellectual capabilities, in contrast to other mollusk classes and most other invertebrate species. Neuroscientists find this intelligence especially fascinating because mammals like humans are so distantly related to these creatures. With around 600 million years since our evolutionary paths diverged, our most recent common ancestor to a cephalopod is something along the lines of a simple worm. This means that they evolved their intelligence entirely independently from us, and discovering insights into the way that their brains work could be really helpful in understanding how brains like ours developed capacity for complex thinking. Some things, like the fact that they have also developed a camera type eye similar to ours, allow for very powerful comparisons to be made between our species. But, for the most part, direct comparison is really tricky. For starters, their nervous systems and brains are incredibly different from ours. For example, the octopus brain is donut-shaped, with their esophagus running through the center. And they have two very large optic lobes on the sides directly next to their eyes. While we have around 86 billion neurons, octopuses only have around 500 million, which is comparable to what we see in dogs. Maybe the most intriguing difference is the fact that over 2/3 of their neurons are actually in their arms. As mammals, we keep most of our computing power in our heads, so one of the current questions in cephalopod neuroscience is, could octopuses be doing more computation in their arms than in their brains to experience sensations and act on their surroundings? Some other burning questions in cephalopod neuroscience are, how does their camouflage system work, both physically and mentally, especially when they only have one particular type of photoreceptor, implying that they only see in grayscale? What evolutionary pressures support this type of complex nervous system in such a short-lived predatory group? If you only live for 2 years and lay hundreds to thousands of eggs, why do you need all of these neurons? How smart are they actually? It's really hard to tell in octopuses because our normal metrics often involve some level of social interaction, and most of their species are cannibalistically antisocial. Cuttlefish, on the other hand, are very social animals, but both groups don't perform all the same behaviors and may not have the same level of intelligence. Also, so many questions about molecular paradigms, which I'm especially excited about. We can't make assumptions about how everything works from a vertebrate perspective. How do their eyes function? And things like tasting with their suckers through chemosensation receptors. Cephalopods have been documented in neuroscience research all the way back to Hodgkin and Huxley's work on the squid giant axon to create their model of the action potential in 1952. Though they have an established history in neuroscience, previous use was limited to exploiting this feature for physiological studies. Interestingly, modern cephalopod neuroscience covers an extensive range of species, often based on local availability and limitations to keeping them in a lab. This variety of species use, while complicating some parts of the research process, is one of the factors that makes this subfield so exciting. With recent successes in applying modern molecular and electrophysiological techniques, as well as a greater understanding of their animal husbandry, there has been considerable interest and growing funding for their use as a comparative model organism in neuroscience.